Howdy, this is Lab Report 12, Phage. This is Group 2, Diego Castillo, and myself, Rita Cantu Mahurin. In the introduction, we highlighted the purpose of the lab, which was to quantify bacteriophage particles using a serial dilution followed by a whole plate titer with E. coli as the host organism. So we prepared a series of 10-fold dilutions and mixed each dilution with E. coli to allow phage infection prior to plating in molten top agar. And then after incubation, the plaques were used to calculate the PFU per milliliter, demonstrating how viral load can be accurately measured through dilution and plating techniques. The materials we used were the phage dilution that was provided by the lab technician, and then we also utilized sterile water, the 9 microcentrifuge tubes, the 4 LB agar plates. We used the micropipette and the tips, and then we also used the 4 large test tubes as well as the molten top agar, which we were able to retrieve from the water bath, which was kept at 45 to 50 degrees Celsius. The cultures we used were the E. coli broth culture, and then we also used our unknown gram-negative organism, which is specific to each group. And we did use the phage stock that was used for the serial dilutions. As far as supplies and equipment, we did use the benchtop vortex, the water bath for the molten top agar, parafilm, the incubator permanent marker for labeling the plates and tubes, as well as our PPE, which included gloves, goggles, and our lab coat.
These are our results for the dilutions. As you can see on the first dilution, the 10 to the negative 3, there's confluent lysis, so the lawn is almost completely cleared and the plaques actually merged. So you can see like there's like the spots and dots that are like really like um, full on the left side that you can't even like tell the difference between some of them. And then on the right side, they're a little more like sparse. But this isn't good enough to see actually how many phages there are because the concentration is way too high and it's not even countable. Um, we use the T4R, which means it's a rapid lysis, so that's why you can see those spots from where the bacteria were just killed and lysed, so you can see the actual spotting. For the 10 to the negative 4 dilution, there's numerous plaques. Some kind of overlap, and they may be borderline countable. There's still a high phage concentration, so it's sometimes usable, but it would be preferred for a less concentration. There's like a big spot in the middle, and I think that was more of an error just on the pouring of the actual media, but that was probably just an issue of slow pouring. It wasn't any factors of any of the phages. And this is our 10 to the negative 5 dilution. You can actually see like specific spots and distinct actual well separated plaques. So like 30 to 200 are like typically seen. Um, this would be pretty ideal to actually tighter the calculation and see how many phages there are here. And then for the 10 to the negative 6 dilution, that one is just not reliable. There's only like 5, 6 um actual spots. So it's very dilute. There's like this confirms the dilution did work, but it's not actually ideal for any counts. Um the purpose of this serial dilution and plating is to reduce that phage concentration stepwise so that at least one plate yields a countable number of plaques, which is that 10 to the negative 5 dilution we were talking about. And that allows an accurate determination of the phage titer. The principle behind this is that each dilution step reduces that phage concentration tenfold. So when plated with that host E. coli and that soft agar, each infectious phage particle produces a plaque, and then clowning those plaques at a known dilution reveals the concentration of phage in the original sample. So, like we said, the 10 to the negative 3 had too much lysis, so there was no distinct plaques. 10 to the negative 4 had dense plaques and sometimes some overlapping. For the 10 to the negative 5, which is the best one we have, it's the clear countable plaques that you can actually count. And then for the 10 to the negative 6, that one had issues with being too little um, phages and it just being hard to see. There was also a bubble that was seen on the media on the 10 to the negative 6 solution. That just happened from when we were pouring. It could have been just too rapid or just too much mixing of the media before we poured it. There's also a spot missing, but other than that, our tests came out pretty great. And to actually calculate the phage titer, you use PFU over milliliters. Then you do plaque count over the volume plated times the dilution factor. So if you had 120 plaques on the 10 to the negative 5 plates and you use zero point 0.2 microliters infection mix was plated, it would be 120 over zero point 0.2 times 10 to the negative 5, meaning that there was 6.0 times 10 to the 7th PFU um, over milliliters. Um, but that's just using an example. That's not actually what ours was. Um, so typically the 10 to the negative 5 is the best one for observing our results. It minimizes error and ensures that statistical reliability. And extreme results include that confluent lysis, where the phage concentration is too high, no plaques, where the phage concentration is either too low or they are inactive. And then there's the uneven lawns, where there's pouring or mixing errors. That would be our 10 to the negative 6 and our 10 to the negative 4 that you can see here, that it just looks wrong. Our best ones that came out were the 10 to the negative 3 and the 10 to the negative 5, where those appear to have no issues with the pouring and the mixing. So that those came out great. And ideally, our 10 to the negative 5 were pretty reliable, and is ideal for counting the actual phages in the sample.
In conclusion, this experiment was important as it demonstrated how serial dilutions and whole plate titers are used to determine the concentration of active bacteriophage particles in a sample. And so by mixing the diluted phage with E. coli and embedding the mixture in the molten top agar, the plaques could form and they represented one infectious phage. And so we were able to calculate the PFU per milliliter from the plaque counts, providing precise quantitative data about the viral concentration, which is essential for virology research and phage therapy applications. Proper technique, accurate pipetting, and rapid plating were critical to ensure that the results were not only reliable, but also interpretable. Thank you so much. This is Group 2, Diego Castillo and Rita Cantu Mahurin.